Dr. GB, and in this video, on the subsequent ones, we are going to talk about the second reason for why the COVID vaccine was developed in a year. And that's because the mRNA technology that was used to design the COVID vaccine is actually pretty old. In fact, it's been in development for nearly 30 years as a proof of concept, and it's been developed as a vaccine potential platform for the past 15 to 20 years. So let's go ahead and learn a little bit about this technology. So the idea that mRNA could be used as a vaccine, so inserted into cells, you make a protein that's on the surface of the virus, and then our body forms an immune response to that protein, was first born in the 1990s by a scientist named Katalin Karikó. Now, she had a lot of trouble getting funding, and the idea didn't catch on super quickly. And the reason being, do you remember how way back in the first series of videos we talked about how RNA has an extra oxygen that DNA does not? Well, this extra oxygen is really important because it causes the cell to attack the mRNA and take it apart really quickly. So what happened is Dr. Karikó just had this idea, and other scientists just kind of blew her off and ignored her because they figured the mRNA would just get chewed up before it could do anything effective. Then about 15 years ago, scientists realized that they could manipulate the mRNA to get it to stay alive in the cell long enough to do its job before eventually getting attacked and broken down by the components of the cell. So specifically what the scientists that could do is they could add special sequences to the ends, or add particular RNA bases to the ends of the mRNA that would protect it, but they also developed like a gift wrap or like a packaging to protect the mRNA from the body until it got to where the same area where the ribosome was. And this packaging was made out of lipids, which is just a fancy word for fats or fatty acids. So around that time, like 15-ish years ago, scientists figured out that you could hide and modify mRNA to get it into the cell, so our cell could take that mRNA, make a protein, and then form a response against that. So once we figured out that this could happen, scientists began to realize that this was a really good potential for a vaccine. And it really became attractive to scientists in the 20 teens, and that's when a lot of the research began.